In this one we are going to talk about proteins. This is part 3. So proteins are the most diverse group of macromolecules, and they have many different functions. The amino acid is the basic unit. There are only 20 kinds of amino acids present in all of life. It doesn't matter if you're an animal or a plant or a bacteria. There's only 20 kinds of amino acids that make you up. Number 2, DNA, or deoxyribonucleic acid, specifies the order in which the amino acids occur in a particular protein. There are various structural levels: primary, secondary, tertiary, and quaternary. And I want to go through those different levels. I know it doesn't leave spots for it in your notes, but it is expected by Glenville for you to understand a little bit of it for this upcoming test. So I'm going to just real briefly go through it, and then when we get into building those proteins and those processes, you will learn a lot more detail about how these things work. So I've built you a couple of amino acids out of those 20. I've built you glycine, and glycine looks like this. We have our carboxyl end of our amino acid. It's got an OH group, a C double bond O. We've got our amine end, which is nitrogen with 2 hydrogens. And we also have our middle carbon, and it's got 1 hydrogen on it. That's always going to be there. And then our functional group. And the functional group for glycine is a single hydrogen. That's what you're looking at. It's just 1 single hydrogen for glycine. Okay, I've also built alanine. And these are the simplest amino acids I could build because these things hate me and fall apart all the time. So I've also built alanine, and I do not know why my computer keeps doing this to me. And it's coming back up in just a second. In 3, 2, 1, I have to add the video back. Just like in class. We always have problems, just like in class. I believe we're still recording. Hopefully we are. But this is alanine. Again, you've got your carboxyl group right here. You've got your amine group right here, which has your nitrogen. And this is my hydrogen off of my middle carbon. But here I've got a CH3 group. It looks kind of like a helicopter. This is my functional group, or my R group, for this amino acid. Okay, so in my primary structure, what happens is I have amino acids that are going to join together in a chain. Think of it kind of like beads on a string. So this is one bead, my glycine is my other bead, and we're going to go through dehydration synthesis again. So what is dehydration synthesis? We are removing a water and forming a bond where those molecules were. So I've got my OH and my H. So what's going to happen is I'm going to remove my oxygen and hydrogen from my carboxyl group, and I'm going to remove my hydrogen from my amine group, and there's my water molecule. And now my amine group needs a bond. Nope, I lost my functional group off of my alanine. I told you these things hate me and are falling apart. And there's my carboxyl group that needs a bond, so it will form a bond like this. And so now I've got my amino acid beads. It's got my carboxyl head, my tail, that's my amine tail, and the next time it bonds with another amino acid, one of my hydrogens will come off of my amine group, and it will bond to the OH group off of a carboxyl. So these just make a long chain in the primary structure. In my secondary structure, hydrogen bonds will create repetitive folds. And this is not between the R groups, this is just between the carboxyl and the amine groups of the chain itself. So it might make a spiral or a beta helix, and every 4 carboxyl groups will make a hydrogen bond with the amine group. So the oxygen from one carboxyl group will make a hydrogen bond with the amine group, and it kind of spirals down. Or it might make an alpha fold, and that's also happens between you have multiple rows of them, but they make those hydrogen bonds as well. So it kind of starts to form a structure here, but it's still not functional. It still can't do any jobs. Next, you have your tertiary structure. Now we're getting, if we were dealing with our letters, or our letter example, our primary was our letters. We made words in our secondary, and our tertiary is we're going to take those words and make sentences. So now we're making our 3D model. So this is when these chains, these alpha folds or these beta helixes, will kind of start to turn and wrap on themselves and begin to make more of a 3D instead of a linear structure. This happens because of interactions between the R groups. So the R groups get a lot more complicated than what I developed. CH3 and just the regular hydrogen is a very, very simple R group. they get very complex and they can branch out a lot. So you can have different interactions between those where R groups are attracted to each other. Sometimes those attractions are hydrophobic attractions, sometimes they are hydrogen bonds, and sometimes they're disulfide. If you think back to the perm lab that we did, those of you who had an AP, we were changing those bonds with the cysteine amino acids. So those are your three-dimensional structures. Now, a lot of proteins only end in tertiary structures, but some of them actually go on and become quaternary structures. And if that's the case, then you have a couple of tertiary proteins where they're making these 3Ds and they kind of fit together in different ways. And so you have more than one amino acid chain joining together. And the things that hold those together are the same kind of interactions that hold the tertiary structures. Those are your R group interactions that are joining those together. So again, they are functional at the tertiary level, which is when they have their three-dimensional structure. Before that, we are just kind of bonding, making hydrogen bonds in between the amino acid groups. I'm not even on the right part of my notes. Apologies, mates. Let's come back down here. There we are. Functions. Let's talk about functions of proteins. The first function is a structural function, such as keratin, which is in your hair or nails, or collagen, which is found in your ligaments. They can be messengers, hormones. Not all hormones are steroids. Some of them are proteins. They can be involved in transport. So hemoglobin transports oxygen in blood. They can also transport stuff across the cell membrane. Aquaporin is a protein in the cell membrane that transports water from one side to the other. And then you have other receptors and parts on the cell membrane that are involved in transport of particles. They can be involved in protection, your antibodies, which fight germs. And remember, you also have antibodies in your blood. If you're type O, type A, or type B, you have those different antibodies. And they can act as enzymes, which speed up chemical reactions. So enzymes are site-specific, which means they have a specific shape, and only the molecules that fit into that shape can act in on them. If you think back to AP, I let you guys watch a video about bacterial communication. I will send out an email with that because I just think it's so cool. But those proteins that are on the cell membrane act as receptors for specific types of chemicals. And you could mimic those and block or send signals based on the shape of the protein. So again, you've got to think of proteins as three-dimensional objects, and based on their structure, that three-dimensional structure, they do whatever function they're designed to do. So that's your section on proteins. Your next section will be on nucleic acids.